Hello, everyone. My name is Jason Stein. I'm an associate professor at UNC Chapel Hill in the Department of Genetics and the UNC Neuroscience Center. And I'm happy to share with you today some of the work we've been doing in my lab on cellular quantification in tissue-cleared whole brain mouse images and human cortical organoids. So causal genetic factors are starting to be identified for different neurological and psychiatric disorders. Identified genetic variants that create risk for a disorder are just starting points in understanding what causes these disorders. Base pair changes in the DNA do not directly alter human behavior. Instead, they must be mediated through some molecular, cellular, and systems level cascade. My lab attempts to make a contribution by understanding how disease-associated genetic variants affect gene regulation, cytoarchitecture, and macroscale brain structure. Much of my previous work has been on mapping genetic effects on macroscale brain structure as measured through MRI. In this type of study, we need a population of genotyped individuals where we have measured their genetic variation, as well as images of macroscale brain structure where we measure variability in brain size. Within the Enigma Consortium, our recent project sought to identify common genetic variants influencing cortical surface area and thickness. We acquired MRIs and genome-wide genotyping from 60 cohorts across the world, comprising almost 34,000 participants, with replication in an additional 17,000 participants. We identified hundreds of loci in the genome where variation is associated with inter-individual differences in cortical structure. But these intriguing findings always leave us with another question, and that is, what is the cellular basis underlying these differences in gross anatomy? Is it changes in the total number of cells in individuals of different genotypes, changes in the number of cells of a given cell type, changes in the overall size of cells or their branching patterns? And unfortunately, MRI does not have sufficient contrast or resolution to answer these questions. So we've recently turned to different techniques. So today, I'm going to talk to you about how our lab is studying how genetic variation influences cortical cytoarchitecture. That is how cells are organized in the brain. And we do this, we measure the brain cytoarchitecture through tissue clearing and light sheet microscopy. So there's been a recent surge in new methods for imaging intact brains at cellular resolution without the need for physical slicing. These are called tissue clearing methods and allow the full mouse brain to be imaged intact. We've imaged a mouse model of neurofibromatosis type 1, a disorder which often presents with intellectual disability and autism, to understand how this mutation alters brain structure to lead to risk for a neuropsychiatric disorder. We've used a mouse model that knocks out the NF1 gene specifically in progenitor cells of the developing cortex using an EMX1 Cree flox design. These mice then have both copies of their NF1 gene disrupted, only one copy of their NF1 gene disrupted, or no copies of their NF1 gene disrupted. We acquired 6 mice in each of the different groups. We then use iDisco tissue clearing technique to be able to image these and immunolabel them with markers of lower layer neurons, which is marked by CTIP2, upper layer neurons marked by CUX1, or all nuclei marked by TOPO3. These were then rapidly imaged on a commercially available light sheet microscope. So this is an example of what a tissue-cleared mouse brain hemisphere looks like. At the top of the image is the rostral side of the brain where the nose is, the lateral side where the ears are, and the back here of the brain, you can see the bright folds of the cerebellum. So this image is quite zoomed out, but labeled with TOPO3, which is a nuclear marker. So here you can see that the brighter the image is, the higher the density of the nuclei. So you can see, like in regions here of the hippocampus, there's a really dense number of nuclei. So zoomed in on the right side here, you can see that these images provide sufficient resolution for visualizing individual nuclei in the brain of a P14 mouse, both in XY and XZ. So as all the neuroimagers at this conference know, analysis of images requires a detailed processing pipeline. So we went over the top 3 here. A graduate student in my lab, Oleg Krupa, who recently graduated, developed a new pipeline for generating and analyzing these images on a commercially available light sheet microscope. This pipeline includes image stitching and alignment across different channels, registration to a defined atlas that is well annotated, as well as quantification of features within the image, which in our case is nuclei, and then to quantify these nuclei and co-localize them with different markers for different cell types. After that, we have maps that show the differences in cell counts between different brain regions here. So here's what these images look like. There we go. With the right side being the NF1 conditional knockout and the left side being control. So here you can see labeling of two different layers of the cortex, the lower layer and the upper layer. We also have a nuclear stain, which we use to count every cell. We're able to count cells within each region of the cortex, which is identified through registration to an annotated atlas. And you can see those registered regions here. And so as it finishes going through the cortex, you can see that it zooms out here in this video. And you can see the NF1 conditional knockout has somewhat of a bigger brain compared to the control. So these images are quite beautiful, but of course we need to count features in the image in order to make quantitative comparisons. So in order to quantify nuclei, we first needed to train a computer what a nucleus looks like. Ole, the grad student leading this project, with the help of some undergraduates in the lab, manually labeled over 50,000 nuclei by hand to train a deep learning model called a UNet. Ole then applied that UNet to the images and was able to quantify all the cortical cells, as shown here. So each dot represents the centroid of a nucleus. The dots can be seen here. And we compared these automated labels to the independent manual labels and found them to be very accurate compared to other methods. So they were compared to two methods, ClearMap and Cubic. What the program calls as a nucleus is usually a nucleus, so it has relatively high precision. Although the program does miss some nuclei in densely packed regions, which is evidenced here by somewhat lower recall. So also the more training that we provide the program, shown by the number of images as input, the better the algorithm does to call a nucleus. So overall, nuclear classification can be done in P14 brains using a deep learning technique. So you can see that this technique can be used to count all the cells in the cortex, as well as to co-localize with markers for different cell types, upper or lower layer neurons. And each dot in this video here represents a different cell type, or sorry, a different cell. And then zooming out, you can see the entirety of the cortex and then labeled quantified nuclei for the upper layer neurons or the lower layer neurons here. So what did we find? So here you can see a representation of a flattened cortex, the nose at the top here, the cerebellum at the back here. So using NuMorph, we measured the average cortical thickness of cortical regions and detected increased thickness in the posterior regions, as well as slightly, we detected increased thickness in the posterior regions and slight cortical thinning in the orbitofrontal regions here. So these differences bear a strikingly similar pattern to human MRI findings, where patients with neurofibromatosis type 1 were shown to have thicker occipital and thinner frontal cortices. Next, we found that the overall increased thickness was associated with increased cell count, as shown by these TOPRO3 here, but not from excitatory neurons, as shown by this CUX1 or CTIP2 positive, instead by non-excitatory neurons, which must be glia. So in sections, we found that there was an increase in astrocytes marked by GFAP and oligodendrocytes marked by OLEG2 that explain this increased thickness. So here we think that we have explained in part the cellular basis underlying MRI findings in human, though it would be good to, of course, to confirm this in postmortem human tissue from neurofibromatosis type 1 patients as well. So overall thickness increases were driven by non-neuronal glial populations. Though the thickness findings are similar to what was found in human, we also were able to explore differences in cell type proportions that would not be visible on MRI. We found a very interesting set of areal differences in the NF1 cortex. There was a depletion in the proportion of upper layer neurons in more medial regions of the NF1 conditional knockout as compared to wild type. There was also a decrease in lower layer neurons in more lateral regions of the NF1 conditional knockout as compared to the wild type. So there seems to be a reciprocal change across the cortex where depletion in the proportion of upper layer neurons is associated with increase in lower layer neurons. We thought that these findings were quite interesting given that slice immunohistochemistry at different regions may give you quite different results. So this emphasizes what we think the utility of the 3D approach of imaging. So since the publication of our article, we've been expanding into several new research directions. So one of them is expanding our training set through manual annotation. Oops. One very important thing to consider is that our algorithm to count nuclei is only as good as the data set it was trained on. So increasing the size and the scope of the training set will be essential to improving counting accuracy, especially in difficult regions like those that are in densely packed areas like the cerebellum, the hippocampus, or embryonic brains. So we have in the lab 15 undergraduates who are now doing this annotation using a tool that's been developed by a visualization engineer, David Borland, who's our collaborator. The goal here is to trace tens of thousands of nuclei in order to have an even better algorithm. We are importantly tracing the entirety of the nucleus here rather than just doing one dot per nucleus. And this will enable us to do instant segmentation. So we have the full shape of each nucleus rather than just counting a nuclei. The tool we developed is called Segmentor, and we call our nuclei labelers nuclei ninjas, with the original thought being that this would be like Fruit Ninja, where you sort of have to separate two nuclei that are falsely attached to each other. So we currently have 14,000 entire nuclei annotated through the dedicated work of our nuclei ninjas and getting more and more every day. Current manual annotation takes a lot of work though for a single user, as each block that we give our undergraduates contains roughly on the order of 1,000 nuclei. So it usually takes an undergrad an entire semester to segment one volume, considering that they usually contribute about 10 hours per week. So our goal here was we have another goal, which is to make it easier to make progress on a given volume, given that we want more users to contribute. So we're hopeful that we can get thousands of manual annotators, but each doing far less work. So maybe just a few minutes of work instead of a whole semester's worth of work. To do this, we need to move away from our standalone application, Segmentor, that I previously showed, and from segmenting the entire volumes. Instead, we need to assign tasks in parallel so many people can be editing a given volume simultaneously, but not fighting each other for annotations. So we're implementing this through a web-based framework that we're calling Ninjato that's now being tested, and we hope we'll be ready for prime time by the end of this year. So the instant segmentation is now being implemented and tested, and we have reasonable precision and recall now, especially for the P14 mice, where we have a larger training data set and where nuclei are spaced farther apart. So we're actively increasing our manual labeling in the P4 mouse to increase performance there. The real advantage, again, of this instant segmentation will be that we can calculate not only the counts of the nuclei, but also features of nuclear shape, like the volume of the nuclei that may be useful for identifying cell types without extra labels. So this work is being done by our collaborators, Gurang Wu and his postdoc, Junbo Ma. Okay, so switching gears a bit, I'm going to talk to you about applying some of these tissue clearing techniques to a different model system of the brain. In this case, human brain organoids, which are 3D cultures of brain cells that mimic some of the structure of the developing cortex. So human cortical organoids can be derived from individuals who are deeply phenotyped. And in this case, we are making cortical organoids from individuals of the Infant Brain Imaging Study, or IBIS, who have had MRI scans and behavioral measurements longitudinally since infancy. It's a very cool and detailed cohort. So from those individuals, we are acquiring blood samples. Isolating peripheral blood mononuclear cells, overexpressing reprogramming factors to turn the cells back in time to an embryonic-like state, which is called an induced pluripotent stem cell. And from there, from the induced pluripotent stem cell or iPSC, differentiating them into cortical organoids, getting whole organoid imaging and being able to relate that back to the MRIs from that same individual to see how well they model the individual's cortical growth. In this way, we're trying to assess the fidelity of organoid cultures. So we generate iPSCs from this population and differentiate them into cortical organoids, which are these little balls of cells. The entire protocol takes around 3 months and involves culture in a mini spinning bioreactor, which spins around. This mini spinning bioreactor is shown here and spins around to increase media diffusion and to prevent cells from dying in the center of the organoid. So after 3 months, there's the spinning. So after 3 months, we fix the cells and again perform the tissue clearing and imaging. We're also getting other measures of these organoids as well, but for today, I'm going to focus on the tissue clearing. So here's an example of an organoid differentiated in our lab. The image of this organoid was obtained using iDisco tissue clearing and light sheet imaging to image the full structure of the human cortical organoid at cellular resolution. We label in different colors markers for progenitor cells, the developing lumen, dividing cells, and all nuclei measured in TOPRO. So here's a video of the organoid, and so spinning around and then going through slices of the organoid. Each of these sections represent neuroepithelial buds, which are like the closure of the neural tube. So each one of those is like sort of a new developing brain. So you can see here a lot of neuroepithelial buds, and these are the expected cell types and morphology of cells, showing that we are indeed generating models of the brain in a dish. So we have applied NuMorph to count cells in organoids, and this seems to work reasonably well for neuronal markers like CTIP2 shown here, and then NuMorph counts here, and CUX1 shown here, and NuMorph counts here. So these label lower layer neurons and upper layer neurons. However, cells that are really densely packed, like the neuroprogenitor marker PAC6, do not seem to be accurately segmentable with our current combination of imaging resolution and our segmentation tools. So for progenitor areas, what we end up doing is measuring the size of the lumen, so marked by NCADherin, surrounding the PAC6 positive cells. So this can be thought of as analogous to the size of the developing germinal zone, which contains the progenitor cells that eventually differentiate to neurons to form the cortex. So we sort of have a two-layered approach here. For neurons, we calculate things differently than we are where we measure neurons, we measure individual cell types, whereas progenitors, we're measuring volumes. We do have some preliminary data here showing that we can correlate the size of the progenitor-like zone in vitro with the overall cortical surface area in vivo. So in a small sample size of only 9 donors right now, who had both MRI at 24 months of age, where the organoids were generated from iPSCs of that individual, we find a relationship between the size of the germinal-like zone in the organoid with the actual size of the in vivo cortex. So you can see that relationship here. This is somewhat expected given that a greater number of progenitor cells in the developing cortex is expected to lead to a larger size of the adult cortex. So far this was only conducted in 9 donors, so we are increasing our sample size for the study. If it holds up, it would show though that organoids are a high fidelity model system and can be predictive about the size of brain structure in the future from that individual. We aim to use this study to understand the cellular basis for brain overgrowth in autism. Okay, so overall, we've developed tools to quantify cellular brain structural differences in 3D images of mouse brains. Using these tools, we found that an NF1 conditional knockout mouse had similar spatial patterns of cortical thickness differences to the human, and that increased thickness is driven by glial cells. We are expanding these studies by increasing our training data set, and we're generating iPSCs and cortical organoids to model cortical development in individuals who have been densely phenotyped from birth. Overall, tissue clearing technologies provide complementary measures to MRI to understand how genetic variation leads to differences in brain structure. So we are applying these tissue clearing technologies in multiple model systems to understand how genetic variation leads to risk for complicated neuropsychiatric disorders. So I really want to thank the people who have contributed and done all of this work. So especially I would like to thank Ole Krupa, who was recently PhD, developed this NuMorph package and worked with our collaborators, Mark Zilka and Lei Jing. I'd also like to thank Rose Glass, who's shown here, who generated all the organoids, and our collaborators from the IBIS consortium, who have allowed us to generate these cortical organoids from their study. So thank you very much for your time and attention.